Okay, so let's start the uh, today discussion regarding the your regarding the course cell biology. Okay, so this is third session of tutorial session. Uh, Many of there is not present, so I don't know what is the reason. But like today, we'll be first discussing the assignment question. We'll be discussing the lectures which was discussed. Okay, in that week, and then we will solve some assignment questions. So previously, like. As I was not aware. Okay, so I had discussed the assignment question which was due. Okay, so assignment questions, the last date was due, but still I had discussed that. But so now what we'll be doing is that the assignment questions which are before the due date, we will not discuss that. Okay, after the when the due date is over, after that only we will see the questions, that assignment questions. Okay, regarding to this session, to this course. So discussing it prior before the due date, it will not be a good strategy, and it will not be a good for you as well. Okay, to attempt a question. So we will discuss that later after the due date. So, but still we will discuss the previous year assignment questions. Okay, with some model questions for regarding this week content, so that you also get to know what are the some probable questions uh, that can be present in that course. Okay, present can be available in the exams if you are attempting the exam. So first we will go through some content, and then regarding this week, revise the content, and then we will discuss the assignment questions, some of them. So share the screen. So is the screen visible to all of you? Is the screen visible to you? Yeah. So this is like third session, and my name is Kundan Solanki. Okay, and I'm a PhD PMR scholar at IIT Indore, and I'm one of your PA of this course. Okay. So in this week, in previous week, basically what we the course the content what we discussed was regarding the cell cycle checkpoints. Okay, and cell cycle inhibition. So cell cycle checkpoints and certain proteins which inhibits the cell cycle checkpoints. Okay, so we discussed over there CDK and cyclins which are present in the uh, CDK and cyclins which are present over the during the cell cycle regulation. So that proteins we discussed, and we discussed regarding the overall cell cycle regulation. Okay, there was like four different phases such as G1, S, G2, and M. Okay, and what are the CDK and cyclins involved over there? We discussed regarding the prokaryotes and the eukaryotes as well. Okay, the different CDK and cyclins which are involved and how it is regulated. Basically, the different stimuli which activates or represses the cyclins and CDK complex. Okay, and the ubiquitin annihilation mediated degradation of cyclin and other factors. Okay, other things which we discussed over there. And we discussed that how are the ACE phase regulated. Okay, the ACE phase is basically important, which helps in the replication of the DNA molecule. Okay, so that thing is important over there. And so yeah, that overall we discussed regarding that CDK cyclins and ubiquitin mediated degradation. So in this lecture, first we will be discussing, as I told, ACE phase is important in the cell cycle regulation. Okay, so how ACE phase is mediating the cell cycle regulation. So we also talked that in the ACE phase, generally the DNA will be forming only at once. Okay, so only once the DNA will be formed, replicate. It is not that multiple times it will get replicated, a single DNA molecule. Okay, so How it is regulated, we will be seeing that. Okay, what are the CDK and cyclins involved and how it is regulated, and the DNA replication after the studying the regulation in the S phase by the CDK and cyclin, we'll be studying the DNA replication. Okay, how the DNA replication occurs, what are the different the initiation, elongation, and termination phase of the DNA replication. We will be studying that in the prokaryotes as well as eukaryotes. So, what are the proteins involved during the replication in the initiation phase, and after that in the during the elongation, what are the protein involved and how it is initiate, how it is elongated, and at the termination, okay, what are the factors which are involved during the termination of the DNA replication, and how the DNA gets terminated. What are the signals which mediates the DNA termination? We'll be discussing that. So today, overall content will be and the content of this week basically for this course was regarding this what was revolved around regarding this. Okay, DNA replication and regulation of DNA replication in the space. So when I talk about the first the DNA replication in A cells, so so that you already know the replication happens in the A phase of the cell cycle after the G1 phase. Okay. And entry into the A phase is highly regulated. As I told ki, uh, the cells will send certain signals and certain stimuli. Okay. It will it will sense the nutrient availability, cell size. Okay. And the presence or absence of mating phenomenon. Okay. When I talk about the budding yeast. Certain nutrient availability and cell size will matter. After that, only the cells will move with the S phase. So there is a nutrient starvation or other inhibitory phenomena which is occurring in the environment, then the cells will not be dividing in that condition. Okay, the cells will arrest into the G1 phase only. Okay, so this is important that the nutrient availability should be there. So the regulatory point is the restriction point in the animal cell and the start point in the yeast. Okay, is crucial in controlling the yeast phase. So if there is a nutrient availability and all the other factors are, if there is nutrient availability and all the factors are appropriate, then only it will pass through the restriction point. Okay, otherwise it will not pass through the restriction point in the cell cycle. And once it is committed from the restriction point, okay, so once the cells moves from the restriction point, there is no way the cells can revert back into the G1 phase. Okay. So then it will follow through completely through the S phase, G2 phase, and M phase. Then the cell has to divide only. Okay. Cell has to replicate its own DNA. It will go through the S phase and it will not revert back into the G1 phase again. So this is why it is important that restriction at the restriction point, all the factors should be regulated properly. Otherwise, we will see in the further also during the cancer. Okay, generally during in the cancer, what we say that the cancer is a unproliferated dividing cells. Okay, so basically the regulation is hampered in the cancer cell. That is why it is uh, unregulated proliferation is occurring. So the one of the steps which is unregulated is this restriction point. Okay, restriction point. So in cancer, basically the factors which regulates the restriction point during the restriction point. Okay, movement of cells from the G1 phase to S phase. That proteins are basically mutated. Okay, or its function is inactivated. That is why the cells does not get regulated during this time point. Okay, and cell doesn't sense the signal that I have to stop the division. Okay, so that is why continuously gets dividing, and that is how the cancer occurs. So this is one of the reason, basically, how why the cancers are, why there is unproliferated cell division in the cancer. Okay, this is a very important step to study. And if there is no appropriate condition is not there, then the cells will move into the G0 phase. Okay, quiescent stage, that is the zero phase. So there are certain key regulators involved in the cell cycle transition. So in yeast, generally there is this CDK1, okay, at the start point, that is G1 stage. So, for example, the cell cycle phases you already know G1S, G2, and M, and there are cyclins which are involved over there. Okay. So, in each generally CDK1 is involved with G1 cycle is CLN1, 2, 3. In A space, CDK1 is involved with the CLB5 and CLB6 S cyclins. And in the mitosis, generally CDK1 is involved in CLB14. So in yeast, generally the only the one CDK is present, that is CDK1. Okay, and cyclins may differs on each other. Okay, at the different stages, but the CDK is common. But in the animal cell, the at the restriction point generally there is CDK4 and CDK6. Okay, at the with the cyclin D. Okay, at the restriction point. So if this cyclin and CDK complex is there, then the cell will move through the restriction point. Okay. If it is not there, then the cell will not move through the restriction point. So basically, you need to regulate the expression of this cyclin D because, as I told in the previous lecture as well, the level of CDK remains constant. Okay. So for example, CDK4, 6, as well as CDK2 and 1 in the G1 and G2 and M phase. Yeah, levels remain constant. They does not get degraded. The degradation occurs of the cyclin only. Okay, that is cyclin DEA. So if you want to inhibit certain checkpoints, then you degrade that cyclin, and you can inhibit the checkpoints. Okay, movement of cell from the one phase to another, because that cyclin will not remain activated. So that is how cell cycle inhibitors. Okay, that is basically cyclin CDK inhibitors. They inhibit the cyclin. Okay, they inhibit the cyclin promotion. Inhibit the cyclin and mediate its degradation so that the cell does not get promoted into the next phase. So cyclins get degraded, CDK does not get degraded. Okay, so in animal cell, in the G1 space, CDK4 and 6 are present, and cyclin D is there. Okay, along with that. So then it moves to the restriction point, and over there, G1 to S transition basically CDK2 and cyclin E is there. Okay, CDK2 and cyclin E is present in the G1 to S checkpoints. And in the S to G2 phase, S and G2 basically CDK2 cyclin A complex is there. Okay. CDK2 cyclin A complex will be there and in S and G2. And G2 to M checkpoint, basically CDK1 and A cyclin will be there. And in progression to M phase, CDK1 and cyclin B. So these are the different cyclins, CDK complex, which are involved at different stages of the cell cycle. And why we need to have different cyclins and CDK complex during different phases of the cell cycle is because then only we can regulate certain checkpoints. Okay, so for example, if the G1 and S cyclins are common, okay, CDK cyclin complex of G1 and S are common, then it will be very difficult to regulate the cell cycle checkpoints. Okay, for example, if you want to inhibit the G1, then we need to inhibit both the cells, both the CDK cyclin complex. It will inhibit the S phase as well. Okay, so different cyclin CDK complex present because then it will be easy to inhibit that cell cycle phase, and that is how the cell cycle can be regulated. Okay, so that is why different cyclins are present. So there is different growth factors which are involved. Okay, which helps in the formation of the cyclin D. So as I told, growth factors basically helps in the activation of the cyclin complex. Okay, and cyclin helps in the this helps in the formation of the CDK cyclin complex. So as I told, that nutrient availability is important to travel the cells from the G1 to S phase. Okay, so through the restriction point. So at the restriction point, cyclin D is present. And cyclin D expression is mediated through growth factors. Okay, so if the nutrient availability will be there, then only cyclin D will be expressed. Otherwise, it will not express. And if the cyclin D is expressed, then only the cells will move through the restriction point, okay, and into the S phase. So growth factors will move through the pathway called RAS, RAF, ERK pathway, and it will synthesize the D cyclin. and D-cycline will bind to the CDK4/6 complex, and it will move through the restriction point. And after movement to the restriction point, the D-cycline will get degraded, and cycline 2E complex will form, and that will then again move through the S phase. Okay, cycline 2, cycline A complex. That is how it is regulated. And here you can see how the external factors activates the cycline D activation, okay, expression. So growth factors bind to this receptor tyrosine kinase. Okay. So growth factor binds and this receptor tyrosine kinase autophosphorylates itself. Okay. So after the phosphorylation of this receptor, these adapter proteins such as GRB2 and SOS will bind. Okay. And this GRB2 SOS will convert GTP into GDP and activates the RAS protein. Okay. So RAS protein will get activated, which is a membrane. RAS protein is a membrane-bound protein, and activation of uh, RAS GTP complex activates RAF protein, okay, which is a mediator of the signaling, and RAF will activate a MAP kinase cascade, and then it will activate the ERK protein, and ERK will then activate the transcription factor and mediates the cyclin D expression. Okay. So that is how the growth factor presence is correlated with the cyclin D expression. And if the nutrient availability will be not be there, then the growth factor will not be present in the medium. Okay, and the cyclin D will not there. And if the cyclin D is not there, then the cell will not move to the restriction point. Okay, so that is how the overall it is regulated. So now there is another level of regulation as well through the cyclin D and CDK4/6 complex. From movement from the G1 phase into the S phase, okay, through the restriction point. As I told that growth factors mediate the cyclin D activation, and cyclin D basically interacts with the CDK4/6 complex, and then the CDK4/6 will phosphorylate the retinoblastoma protein. So retinoblastoma is a protein which inhibits the E2F transcription factor. Okay, it associates with the HTAC also. So the role of E2F transcription factor is to activate the E2F transcription related gene, that is S phase genes. Okay, so S phase genes involve cyclin E and cyclin A. So cyclin E and cyclin A are the cyclins which are present during the G1S CDK, G1S and S phase respectively. Okay, so cyclin E will bind to the CDK2 complex during the G1S CDK, and cyclin A will bind to CDK2 during the S CDK phase. Okay, so it is if you want to stimulate the cells and move through the S phase, then this E2F should be active, so that only the cyclin E and A will get transcribed and activate the CDK cyclin complex. Okay, so for that you need to remove this retinoblastoma protein from this transcription site. Okay, that is then only the E2F will get activated. So that process is mediated through CDK4/6 cyclin D complex. So that it phosphorylates the retinoblastoma protein, and phosphorylation of retinoblastoma basically removes this retinoblastoma from the E2F transcription site. Okay, it translocates this retinoblastoma outside of the nucleus, and it dissociates the E2F and retinoblastoma complex. So now E2F is free, and this E2F mediates the transcription of S-phase genes, and these S-phase genes such as cyclin E and A are transcribed and forms a complex with the CDK2. Okay, and further, the CDK2 cyclin E as well as CDK2 cyclin A complex as well mediates the over phosphorylation of retinoblastoma. Okay, so even if the after the movement of cells from the G1 CDK to the S CDK phase, okay, S phase basically movement of cell from the restriction point through the S phase, then also the retinoblastoma protein will remain phosphorylated. Okay, because the CDK2 cyclin E and cyclin A complex will also phosphorylate additional retinoblastoma protein, so that the E2F remains constitutively active during the whole S phase genes. Okay. After completion of the S phase, the cyclin E and A will also get degraded. Okay, and that degradation will then remove the phosphorylation of the retinoblastoma protein, and then the retinoblastoma can again bind to the E2F. So if the cells move from the S phase to the G2 phase, okay, so during that time we don't need the S phase genes. Okay, when the cells pass through the S phase and move to the G2 phase, so during that time E2F should not be active. So during at that how it can be how it is regulated, the retinoblastoma will get dephosphorylated and then it binds again to this E2F factor, and then it will inhibit the transcription. Okay, so phosphorylation will not be occur because cyclin E and A will not be present during the G2 phase, as well as cyclin D will also not be present. Okay, because it will be degraded at that phase. So that is how this is regulated in the cells. Okay, the restriction point. So is it clear to everyone? Okay, so there is like certain restriction points, certain inhibitors of this CDK cyclin complex which are present in the cell cycle. So during that, if the CDK cyclin complex are inhibited, then the cells will not move through the cell cycle. Okay, so for example, INC4 family, which contains this P15, P16, P18, and P19. Okay, so these are the inhibitors of this CDK cyclin complex. Okay, CDK4, CDK6, and G1 and G1 phase. So INC family P15, 16, 18, and 19 will inhibit the S phase, G1 phase of cyclin CDK complex, and CIP and KIP family, that is P21, P27, and P57. So we inhibit the CDK2 cyclin E and CDK2 cyclin A complex, that is G1 and S and G2 phase. So P27 and P21 will inhibit the CDK1 cyclin B complex. Okay, CDK1 cyclin A complex basically. Okay, so if we inhibit the CDK cyclin complex, then the cells will not move through that phase. Okay, and that is also one of the mechanism of regulation, that if these inhibitors are present, generally during the cell cycle division, okay, and activation, these inhibitors are not present. Okay, these inhibitors are not present. But if there is a certain condition, okay, during which the cells need to stop the regulation, okay, stop the replication process and division process, then these inhibitors will be activated, and that will inhibit the CDK cyclin complex present in that cell cycle phase, and then this will inhibit the cell cycle progression. Okay, so these are the some of the inhibitors. So as I told that this deregulation of the cell cycle in the cancer. Okay, so cyclin D is a critical factor as we discussed. Okay, and it defects in this result in the loss of regulation. So as I told that unregulated expression of cyclin D contributes to cancers. Okay, mutation that causes unregulated expression of cyclin D. It contributes to the lymphoma and the breast cancer. And mutations that inactivate the that CKIs, okay, INC4 CKIs, that is the inhibitors of the cyclin, okay, that binds and inhibit the CDK4 cyclin complex are also common in cancer. So if the inhibitors are mutated, okay, and if it is inactivated, then this CDK4 cyclin complex will remain constitutively active. And this if remains constitutively active, then the replication will continuously occur. Okay, so the role of these inhibitors are to uh, limit the activity of the CDK cyclin complex. Okay, so that the cells does not continuously divide. And if it is inactivated, then the cells will remain continuously dividing, and that is found in commonly in the cancer. And also the as we discussed regarding the retinoblastoma protein. Okay, so retinoblastoma are the proteins as we discussed that if it is phosphorylated, then it becomes inactivated, and that mediates the uh, transcription of the S phase genes and the cell cycle division. So retinoblastoma basically is a tumor suppressor gene. Okay, so if it is active, then it will limit the growth of the tumor. That is, it will inhibit the cell cycle division. Okay, cell cycle replication. But if it is inactive due to CDK cycline complex by phosphorylation mechanism, that will lead to the replication of the DNA and uncontrolled proliferation of the cell. Okay, so tumor suppressor are the genes which suppresses the replication during the when it is active. Okay, so when it is inactive, then it will promote the proliferation. So that is called tumor suppressor gene. Another example of tumor suppressor gene is your P53. Okay, P53 also, if there is a DNA damage, as we discussed in the previous tutorial session. Okay, if there is a DNA damage, then P53 will get activated, and the P53 get activated, and it binds to the DNA place at which the DNA it binds to it acts to the transcription factor basically, and helps in the transcription of P21 inhibitor. Okay, so here we can see P21 protein, which is an inhibitor of CDK1 cyclin B complex. Okay, so here also you can see P21 is a CDK2 cyclin E complex inhibitor. So this P21 transcription by P53 inhibits this CDK cyclin complex, and it will not allow the replication of the DNA. Okay, after fixing of the DNA damage, then only this cyclin CDK complex will get activated. Okay, so during that phase, this inhibitor will get degraded, and this CDK2 cyclinic complex will get activated. Otherwise, it will not remain activated. Okay, so this is several mechanisms, regulatory mechanisms, which occurs during the cell cycle division. Yeah. So uh, is it clear to everyone? Basically, up to this point. Hello. Yeah. So now we will move through licensing of the origin of replication. Okay, licensing through the ORI side. So now we will slightly move towards the DNA replication part. Okay, so how the DNA is getting replicated and what are the initial phase of the DNA replication, what are the protein involved over there, and now we will go through that part. Okay, now the cell cycle, DNA replication stage of the cell cycle. So the initiation of the replication is divided into two steps basically. Okay, so there is this origin of replication site, ORI site. Okay, so at this site only the replication will be initiated. Okay, and not the other sites. So origin of replication is a site on which the replication proteins, okay, the proteins which are involved in the replication binds, and it initiates the replication at this point. So in the late M and G1 phase of the cell cycle, that is early G1 phase of the cell cycle, the MCM complex, that is replicative helicase, okay, MCM pair of MCM binds to the this origin, forming the pre-replicative complex, okay, pre-RC complex, and this is termed as the licensing of the replication. Okay, so there is a origin site, okay. So yeah, for example, this is your DNA. This is your origin site, okay. And at the origin site, basically, at the early G1 phase or the late M phase, the helicase will bind. Okay, so role of the helicase is to unwind the DNA. Okay, so to unwind the DNA, basically, the helicase needs to bind to the origin of replication. So MCM helicase will bind over here. Okay, MCM helicase will bind and it forms the pre-RC complex. Okay, over here. It forms a pre-RC complex. And this process is called licensing. Okay, this process is called licensing of the replication. And initiation can occur only on this origin only. Okay, so first process is licensing of the ORI. Helicase will be present and then certain protein will get activated. This that activates the DNA helicase. Then only during the S phase only the helicase will get activated and it will move through the cell cycle, move through the DNA replication. In the S phase, DNA helicase are activated by certain proteins which will be seen. Okay, and results in the DNA unwinding and along with the DNA synthesis. Okay, by DNA polymerase. And the DNA and the helicase move through the ORI site along with the replication fork. Okay, so replication fork will be formed. The replication fork is basically this will be the unwinding of the DNA after the activation. Okay, after the activation, the DNA will get unwound and the replication fork will be present at this 2 point. Okay, so over at this site and around this site, both the side, the DNA will get replicated by the leading and lagging strand. Whether that is how the DNA will move through the both the direction, and this will complete the overall replication process of the cells in the cells. Okay, and the uh, origin site does not now lacks the helicase. Okay, so once this helicase is activated, this helicase move through the DNA and it will unwind the DNA and the DNA polymerase will polymerize the DNA. Okay, the new strand of the DNA polymerase. New strand will be polymerized by the DNA polymerase. Okay, and because the helicase will remain activated and it moves through, the origins can only be activated only once per cell. Okay, so it will not be that after the origin. Okay, so once the helicase gets activated and move through the DNA, another MCM complex and another MCM complex does not bind to this newly formed DNA. Okay, MCM complex will only bind after the M phase and early G1 phase. During the S phase and G2 phase, the MCM complex will not bind again to this ORIC. That is how the origins is regulated. Okay, the DNA replication is regulated at that is at this point because it does not allow the new MCM complex to bind again to the newly formed DNA. It will only bind after the division. Okay, after the division of this two daughter strand. So that is how it is regulated. The process of replication. So now regulation of the initiation, as I told that at the origin replication complex, ORC protein will bind. So this is the ORC protein that will remain bound throughout the cell cycle. Okay, so throughout the cell cycle, the ORC will remain bounded to the ORI sequence, origin replication sequence. Okay, origin site, the ORC will be bounded, and this ORC basically helps in recruitment of other proteins at this or at the origin site. So basically, it helps in the identification of the origin site by the other protein, such as MCM helicase. Okay, so CDC6 first binds to this ORC site, and CDC6 is present at low level during most of the cell cycle, and during the early G1 phase, the CDC6 level rises. Okay, and CDC6 if during the early G1 phase, basically the CDC level of CDC6 remains activated. And CDC6 level, if the expression is higher, then it recruits the MCM helicase. Okay, so MCM helicase is recruited by the CDT1 complex. Okay, CDT1 recruits the MCS helicase in front of ORC. Okay, in adjacent to the ORC, the CDC, the MCM helicase gets recruited. Okay, and then the CDT1 and CDC6 complex get dissociated, and MCM helicase gets loaded onto the ORC complex. So this complex is called pre-replicative complex. Okay, pre-RC complex, which constitute the ORC and the MCM helicase. So this is called pre-replicative complex. So now this is during the initiation phase of the DNA replication. Okay. Now this origin is licensed. So this origin This is not the activation. Okay, the activation signals are coming afterwards during the end of the G1 phase. Okay, during the G1 S phase transition. Okay, at that time the activation signal will be get activated. Okay, and disactivate the MCM helicase one bound. But first the MCM helicase needs to be loaded. So that occurs during the G1 phase. Okay, late M and G1 phase. So this is how the licensing occurs. And once the initiation starts of the replication, okay, during the S phase, so now the replication needs to be initiated, okay. So during that time point, the SCDK complex stimulates the assembly of several initiator proteins on the helicase. So initiator proteins are like proteins which helps in the activation of this MCM helicase, okay. So MCM helicase will get activated by certain initiator proteins, okay, and that triggers the initiation of the replication. So, protein kinase known as DDK. So, DDK is a kinase which helps in the activation of the DNA helicase, that is MCM helicase. It phosphorylates, DDK phosphorylates MCM helicase and triggers the activation of this MCM helicase. So, MCM helicase will now move away from the ORC, origin replication complex, and it helps it move through the 5 prime to 3 prime direction. Okay, towards and it leads to the unwinding of the DNA helicase, DNA. Okay, so that the newly strand DNA will be formed at both the sides. So MCM helicase will be activated by the DTK complex. Okay, and this will then trigger the activation of the replication, and the DNA polymerase will be get recruited during the site of the replication. Okay, so this is called replication fork. So the site at which the helicase unwinds the DNA, that is called replication fork. Okay, and at the replication fork, the DNA polymerase will be kept present to form a newly synthesized DNA. Okay, so this is how the activation of the DNA, initiation of the DNA will be occurring. So, uh, so DNA replication occurs during once per cell cycle. Okay, in the S phase only, in the S phase of the cell cycle. So, as I told that if the DNA replication occurs simultaneously, okay, in a single S phase cycle, there are two, three replications. Then the DNA will not be getting segregated properly. Okay, the DNA replication it will hamper the process. Okay, replication. So how it is regulated basically DNA replication at the once per cell cycle, how it is regulated. So that we will be seeing. Okay. So SCDK inactivates the pre-RC component of the ORC and CDC6 and prevent reformation of the new pre-RC till the end of mitosis. So SCDK that is CDK2 cyclin A complex. Okay, cyclin E complex. So that inactivates the pre-RC, ORC and CDC6 complex. Okay, so that ORC will not bind to that origin replication complex again. Okay, during the S phase. So SCDK will inhibit this ORC and CDC6 binding to the origin site. And if this ORC is not bounded to this origin site, then the MCM complex will not get recruited. Okay, at this site. So only if the ORC will be bounded, then only the MCM complex will get recruited. So SCDK ensures that ORC does not remain bounded, and it remains bounded after the end of the mitosis only. Okay, after the cell division, again the ORC will bind to that site, and then it will recruit the MCM. Okay, so this is how the SCDK regulates the division, DNA replication. And apart from that, SCDK also dissociates CDC6, okay, from the origin. So as I told that CDC6 is important. Important, okay, which plays a role in the recruitment of MCM helicase by the help of CDT1. So if CDC6 is not there, then MCM helicase will not will not come. Okay, an MCM helicase, and then it will not form the pre-replicative complex. Okay, needed for the replication. So ACDK ensures that the CDC6 gets dissociated from the origin and is degraded by the ubiquitin mechanism. Okay, and the CDC6 is degraded by the proteasome. Okay, and it also ACDK also ensures that the MCM proteins gets phosphorylated, and this phosphorylated MCM complex triggers their export from the nucleus. So the nucleus MCM protein will be there. Okay, and phosphorylation of this MCM protein triggers their export from the nucleus and into the cytoplasm, and that is how the DNA replication can also get it inhibited. Okay, so if the MCM will not be present, then the helicase, the DNA will not get unwounded. Okay, and then the DNA polymerase will not act on the DNA. So that is why the regulation on the MCM protein is also important. Okay, and then this MCM protein, phosphorylation of MCM protein is unphosphorylated at the MCDK complex. Okay, during the mitosis, end of the mitosis, the MCM proteins again translocate into the nucleus, and then it will license the origin again. Okay, so that is how it will perform the action. And also the CDT1, which is the recruiter of this MCM protein. Okay, so CDT1, as we discussed, MCM complex was present with the CDT1 complex. CDT1 recruits MCM to the ORC complex. Okay, so during the G1 phase, late G1 phase. Okay, so CDT1 will be present. Okay, and during the age phase, CDT1 will be inactivated. Okay, and if the CDT1 will get inactivated, then it will not recruit the MCM to the site of that complex. Okay, so the pre-RC formation is prevented from the S phase until the mitosis. Okay, so after the mitosis only, this MCM and CDC6 and ORC will get activated, and then only the replication will occur. Before that, it will not form. So this is how there is a fine-tuning regulation present. So this is the summarized form. The limit of the DNA replication, that ORC will bind to the origin of replication. Okay, throughout the cell cycle. Okay, and CDC6 basically the level rises in the G1 phase, and it binds to the ORC. So binding of CDC6 with the ORC will recruit the MCM protein. Otherwise, if the CDC6 is not present, then MCM will not get recruited. Okay, and the for activation of SCDK mediates the phosphorylation of ORC. Okay, and CDC6 dissociate from the ORC origin and disassembly of pre-RC complex. So once the MCM complex and helicase, the DNA gets replicated properly. Okay, and the MCM complex are formed and activated during the end of the S phase. The SCDK will phosphorylate ORC and CDC6, and it dissociates from the pre-RC complex. Okay, and CDC6 will get phosphorylated and ubiquitinated by the proteasome mechanism, and MCM will get phosphorylated and exported from the nucleus. Okay, so during the end of the S phase, and that is how it prevents the re-replication of the DNA. Okay, and during the end of the mitosis, okay, during the start of the G1 phase, the CDK activity reduces, and CDC6 and MCM gets dephosphorylated again, so that it binds to the ORC, and pre-replicative complex forms, which gets activated during the start of the S phase, and again the DNA gets replicated at this point. Okay, so this is how overall this is regulated by MCM protein. Is it clear to everyone? Overall, the process is clear. Hello, is anyone there in the lecture? Okay, so So now we'll be moving through the DNA replication. Okay. So let's take 5 minute break. Okay. After that, we will discuss about this DNA replication. Okay. In the DNA replication process in the prokaryotes and eukaryotes, and then we will be moving further. Okay. We'll discuss assignment question. Okay. So let's start at 4 p.m. Okay. Okay, so let's start. Yeah. So now we'll be discussing about the DNet application. Okay. So we discussed about the airspace cycling regulation and all those things. So now we will discuss in detail in prokaryotes and eukaryotes how the DNA is getting replicated. So DNA, you already know the structures of the DNA and how the DNA strands are formed. Okay. The DNTPs are the nucleotides which are used, and primer template junctions is required over there. Okay. So RNA primers are there, and there the template is formed. The DNA replication occurs in the 3' to 5' direction. Okay. So the movement of DNA will be forming from 3' to 5'. DNA polymerase will incorporate the bases to the 3' OH and it will synthesize the 5'. The phosphodiester bonds are formed by the 3' OH of the already present nucleotide and the 5' phosphate of the incoming nucleotide. So that is how the phosphodiester bonds are formed. Okay, and the DNA is replicated. So this is the model of the DNA replication. So first, there is a leading strand and lagging strand. So in the leading strand basically helps continuously replicates the DNA molecule. In the lagging strand, there is a formation of short segment of DNA molecule. It synthesizes in the short segment. Okay, so that is called Okazaki fragment. So the leading strand synthesize one primer, and then your synthesis of primer helps in the recruitment of DNA polymerase, and it synthesizes the DNA. Okay, in the lagging strand, basically the primer will get synthesized at the replication fork. Okay, so replication fork during at the replication fork, the primer will get synthesized, and then the DNA polymerase will be coming and mediating the synthesis of this Okazaki fragment. And during the end of this Okazaki fragment, uh, there will be a there will be a gap. Okay, so then primer will be removed. Okay, after the DNA replication, the primer will get removed from here, and the DNA will get synthesized by another DNA polymerase, and the gap will be filled by DNA ligase. That is how the Okazaki fragment synthesis happens. And the formation of addition of primers occurs at the replication fork only. Okay, so at the replication fork, the addition of primer will be formed. So certain like in the lagging strand, 100 to yes. I'm sorry. Yeah. So the uh, replication fork generally Okazaki fragments are formed, and that is 100 to 2000 nucleotides. Okay. Uh, at the sequence. So in the eukaryotes, generally the Okazaki fragments are smaller, that is 100 to 400 nucleotides. And in the prokaryotes, the Okazaki fragments are larger, that is 1000 to 2000 nucleotides. Okay, so these are the things. And the Okazaki fragments are generally formed near to the leading strand synthesis. Okay, are a sequential order. At the replication fork only the primer will be synthesized, and the leading and lagging strand synthesis will occur simultaneously in the opposite direction. Okay, so DNA replications move anti-parallel to each other. Replication of DNA, it is not parallel. So now, so this is the bidirectional mode of replication of the DNA. Okay, so in the bidirectional mode, for basically in the one strand itself, the leading and lagging strand will be present. So this is, for example, this is if we divide from here. Okay, this is one origin of replication. Okay, ORI site, and ORC will be bounded, and it will recruit the MCM helicase. So as I told that MCM helicase are bounded in pairs. Okay, so one pair will bound to the left hand side of the ORC, and another pair will bound to the right hand side of the ORC complex, and then it will unwound the DNA in the anti-parallel direction. Okay, in the opposite direction it will unwound. So at this prime there will be a leading strand, and opposite to that there will be lagging strand formation. And simultaneously, in the left hand side, there will be leading strand formation at the bottom and the lagging strand formation at the top. So one thing you need to remember over here is that the orientation of the leading and lagging strand. Okay. So orientation, how you will be deciding that where the leading and lagging strand will be formed. Okay. So basically, this is the 3' end of the DNA, and this is your 5' end. Okay. And this is your ORC. Okay, original replication complex. So this will be your 5' end. Okay, and this is will be your 3' end over here. So the over here you can see 5' end. Okay, so at the from the 5' end to the 3' end, the leading strand will be synthesized. So here it is 3' end. Okay, and over here at the opposite there will be a 5' end synthesis. So here you can see 5'. So over here the leading strand synthesis will occur. Okay, so in this direction, so that it will incorporate the 3' end. And I'm just telling you to remember only, okay, how you will be remembering the leading and lagging strand, the synthesis. So here you can see that 3'. Okay, so 3', that means the 3' should be opposite of this leading strand. Okay, so the lagging strand will be formed in this direction because the 3' will be opposite in direction. Here you can see 5', so that the leading strand will be there will be find the leading strand, so that the 3' end comes in the leading strand formation. So 5' and 3' are opposite to each other. Okay, so that is how you can remember. Over here, for example, here is a 3' end. Okay, so in this case, here is 3' end. So the opposite will be 5'. So 5' end will be present over here. Okay, so 5' will be present. That means the lagging strand will be synthesized in this direction. Okay, in this direction, the lagging strand will be synthesized. And the 3' end, the leading strand will be synthesized in this direction because here you will be finding the 3'. Okay, so that you can identify how the leading and lagging strand will be forming. So at the right hand side, basically the leading strand will be at the top, and in the left hand side, the leading strand will be at the bottom. Okay. So DNA replication basically happens by the origin of replication. At the origin of replication complex, okay, the initiator site, and the helicase and all the other proteins are recruited in that region. Okay, helicase unwinds the DNA and it recruits the polymerase. And here you can see the helicase, okay, which unwinds the DNA, and polymerase mediates the replication. And after that, during the end of the replication, both the daughter strands are formed and the two daughter cells are formed, DNA molecules are formed, okay, two daughter cells. At the origin of replication site as well, certain proteins are involved, okay. ORI sequence basically contains an AT rich region. So why AT rich? Because AT has a two hydrogen bonds, okay, compared to GC, which has a three hydrogen bonds between them. So dissolving the AT region is less energetic compared to the GC rich region. Okay, so that is why at the origin site, certain AT rich regions are present upon which the proteins bind and mediates the release unwinding of the DNA fragment at that region. Okay, so that is the role of the protein initiator proteins, which we'll be seeing. And then the DNA polymerase and all will synthesize the DNA molecule. So replication basically in the prokaryote there is initiator protein such as DNA-A which binds to this origin DNA, okay, and melts the AT-rich region. So in the ORI region, there will be AT-rich region, and the DNA-A will bind to that region and will melt that and leads to the dissociation. DNA-B unwinds the DNA and generate the single-stranded DNA template, and DNA-G, that is a primase, synthesize the RNA primer for the DNA synthesis. Okay. So primase DNA-G, that is in prokaryotes, helps in the synthesis of RNA primer. And from that, the leading strand synthesis and lagging strand synthesis occurs, okay, by the DNA polymerase. And DNA base helps in the unwinding. So initial unwinding of the DNA, initially unwinding of the DNA is mediated by the DNA-A, slight unwinding, and it recruits the DNA-B, okay. Then DNA-C basically helps in recruitment of the DNA-B, okay, L1BT. Then Then it further unwinds the DNA and mediates the activation of the DNA replication by formation of the primers and activation of DNA polymerase. So DNA polymerase are basically a hollow enzyme type of complex which contains several subunits present inside it. Okay, so this is the structure of DNA hollow enzyme, a DNA pol 3 hollow enzyme. So the first thing, the clamp loader. Okay, so clamp loader is this yellow portion you can see, yellow part. So clamp loader is loads the sliding clamp on the primer template duplex. Okay, clamp loader helps in the sliding clamp loading. So basically, the sliding clamp helps in the uh, binds to the DNA polymerase and helps in the processivity of this DNA polymerase. Okay, and it basically it holds the DNA fragment intact. Okay, so it holds the DNA fragment intactly, and so that the DNA polymerase can work on it efficiently. Okay, otherwise the DNA is not wound intactly, then the DNA polymerase will get loosen from the DNA fragment. So that is the role of this sliding clamp. It helps in the processivity of all three core enzyme. Okay, by mediating the replication. So the sliding clamp loader is also bounded by this tau protein, and tau protein are the linker kind of thing. Okay, which connects this with the DNA polymerase 3. Okay, so there are three subunits of the DNA pol 3 core enzyme, and it are bounded by the tau protein present on the sliding clamp loader. Okay, so these are the information. And DNA pol 3 basically helps in the DNA synthesis, and sliding clamp clamps the pol 3 core and enhances processivity as well. Okay, so and singles there are single stranded binding protein which are the protein which binds to the single strand. So in the replication fork basically. Okay, so for example, this is a replication fork, and once there is a leading strand synthesis in this direction, and there is a lagging strand synthesis. Okay, so during the lagging strand synthesis, during the unwinding of the DNA, the single stranded, there will be a portion during which the DNA will be single stranded. Okay, so the single stranded binding protein will bind to the single stranded DNA molecule and helps, and it prevents the rewinding of this DNA molecule. Okay, the parent DNA molecule again uh, to it. Okay, so this single strand binding protein prevents the religation of the parent molecule. Uh, okay, and once the DNA polymerase replicates this fragment, then this DNA ligase, the single strand binding protein, are released. Okay, and after that it can be created. So that is how it is regulated. And DNA pol 1, so pol 3 are the core enzyme which help in the replication, and DNA pol 1 are the help in the primal removal and DNA repair process. Okay, DNA polymerase 1. So in the lagging strand, you need to remove the primer. Okay, after the formation of the Okazaki fragment. So that is done by the DNA polymerase 1. They remove the primer and it repairs the DNA as well. Okay, if there is any mutation. Okay, polymer mutation present in the DNA. And ligates the Okazaki fragments. Okay, and there is topoisomerase, an enzyme, okay, which prevents the DNA tangling during the replication and removes supercoil. So during the replication process, uh, as the as this is unwinding, okay, the DNA is getting unwounded or at this position. So there will be a positive supercoiling in this case. So this positive supercoiling during the further unwinding, this supercoiling will get increased. So this topoisomerase one, it helps in the unwounding of this DNA polymerase. Okay, unwounding of this DNA. So it promotes a negative supercoiling. Positive supercoiling means more and more unwounding. Negative supercoiling means release of the unwound DNA. Okay, so this topoisomerase helps in that regard, so that the DNA remains unwounded further during the replication process. So this is the key attributes of the DNA polymerase. That is, DNA polymerase synthesizes DNA by adding the nucleotide to the 3' OH. and is a single active site that catalyzes DNA synthesis. Okay, and the processivity is like 1,000 nucleotide per second, and has a 3' to 5' proofreading exonuclease activity that removes the unpaired base pair. Okay, DNA synthesis occur from the 3' to 5'. Okay, and the DNA polymerase has an proofreading mechanism as well, so that if the incorrect base are incorporated, then also it will remove that incorrect base. Okay, uh, by uh, by the proofreading mechanism. So this is the ORI sequence in the E. coli in the bacteria prokaryotes. So here you can see there are three 13-mer sequence and five 9-mer sequence. So complete this is 245 base pair, and there is five 9-mer sequence and three 13-mer sequence. So DNA A, which is an initiator protein which helps in the unwinding of the DNA, it binds to this 9-mer sequence. Okay, this 9-mer sequence it will bind, and it allows the so in the 13-mer sequence basically there is an AT rich region. DNA A binding with this 9-mer sequence wounds the DNA, and this allows the 13-mer sequence to open up because it is a AT reach. So it DNA basically creates tension by binding to this 9-mer sequence. Okay, it will bind and then comes together. Okay, each and every DNA A comes together, and this whole complex will be wounded. Okay, completely wounded. So this wounded structure will then create a pressure on this 13-mer. Okay, 13-mer sequence. Because of the pressure, this 13-mer sequence, it will get unwounded due to pressure, positive pressure occurring by this DNA bounded by the 9-mer sequence. Because this is an AT-rich region, the energy to release this hydrogen bond will also be lower compared to GC. Okay, so this is how the DNA will unwound the DNA molecule, basically. Okay, so the RF factors, the replication for progresses at around 500 to 1,000 nucleotide per second. Okay, and DNA basically helps in the origin replication and stretching of the single stranded DNA nearby. Okay, and helicase then binds to the site. Helicase then binds to this site, and then it will recruit the primase, and it will create the primosome complex. Okay, the helicase and primase complex is called primosome. We synthesize an RNA primer to initiate the DNA synthesis. So that is how DNA will get synthesized. So these are the steps. DNA A will capture the single-stranded DNA, and DNA helicase again will bind to the complex that is in helicase. Okay, DNA B with the help of the DNA C. Okay, so DNA C is the DNA helicase loader. So DNA C will so first, for example, here is DNA A. Okay, so DNA A will bind. Okay, DNA A will bind and disrupt. This creates the loop formation. So at this loop, DNA B will bind. Okay, DNA B will bind along with C, and it will help. This is an helicase, so it will again unwound the DNA further. And by unwinding the DNA, it will also recruit polymerase over here. Okay, so polymerase will be recruited and DNA polymerase, and then it will further move the uh, replication process. Okay, so this is how it is done. So unwinding is carried out by the DNA B coupled by the 5 to 3' translocation, and there is single-stranded binding protein as well present, okay, which binds to the single-stranded region in the duplex DNA, and it prevents the single-stranded. It prevents the self-annealing of the daughter of the parent DNA molecule, both the strands. Okay, so this is how this process is occurring. And here you can see like sliding clamp loader and sliding clamp. Okay, so the sliding clamp basically binds to the primer template junction. Okay, so in the DNA, as I told that there will be primer in the Okazaki fragment. Okay, so primer template junction. This sliding clamp will load this primer template around here. Okay, around this position, primer template, it will move further. So at the 3' edge of the primer, it will release the sliding clamp, and again the DNA polymer, it will recruit the DNA polymerase and bind. This sliding clamp and sliding clamp loader will recognize the primer template junction, and at the end of the 3' end of the primer, the sliding clamp loader will dissociate the sliding clamp. Okay, then only the sliding clamp will remain, and this sliding clamp will recruit the DNA polymerase. The sliding clamp loader will be released from the sliding clamp. Okay, so this is how actually it recognizes the primer template junction. So it recognizes that from where the DNA is to be activated. Okay, where from where the replication is to be activated. Otherwise, if the sliding clamp is not recognizing the primer, then this sliding clamp loader and sliding clamp can bind to any region of the DNA. It will replicate from any other region. Okay, so specifically, it needs to identify the primer template junction only. Okay, otherwise it will not. The DNA replication will be random in this case. The DNA polymerase 3 basically is present in the E. coli and helps in the main is the replicative enzyme in the E. coli. So DNA polymerase is a hollow enzyme containing both three core polymerase. Okay, as we seen, like there was one sliding clamp over here, and tau protein was attached. Okay, and around this region, the DNA polymerase was present, and over here the sliding clamp was present. Okay, so this was sliding clamp, and this is DNA pol. This was DNA polymerase, and this is tau protein, which is a linker between the sliding clamp loader. Okay, so this was the structure of the DNA polymerase 3. These steps I already discussed. Okay, so these were the total steps in the E. coli genome, E. coli replication. The formation after the recruitment of the sliding clamp, the new Okazaki fragment will be formed. Okay, and the sliding clamp is assembled in the each primer, and the leading strand is synthesized. Okay, in that case, and the process will be continued in this case. So here you can see in this diagram. Uh, this is a 5' and this is a lagging strand synthesis. Okay, this is a primer, 5' primer, and this is a whole DNA polymerase 3 hollow enzyme. So this is helicase, okay, helicase protein DNA B, and this is a primase. So as I told that helicase and primase forms a primosome complex. Okay, this is a primosome complex, and DNA RNA primers will only be synthesized near to the helicase region. Okay, during the unwinding, so helicase will unwind the DNA, and RNA primer will synthesize in this region. And the sliding clamp loader, that's a hollow enzyme, basically moves along with this helicase. So the DNA polymerase 3, this enzyme, okay, one of the subunit of the DNA polymerase binds to this helicase. Okay, slightly remains in the contact with the helicase, so that if the helicase move further apart, then the DNA pol 3 hollow enzyme also moves in that direction at that speed only. Okay. Otherwise, if this complex remains back, then the replication there will be a problem in the replication. Okay, more of the helicase will keeps on unwinding, and the replication will be the again the duplex will be formed. Okay, so once this helicase moves ahead and the duplex is formed after that, then there is no way the strand will unwind again. Okay, so that is why the DNA pole 3 has to move along with the DNA helicase, so that during the unwinding itself, the replication needs to be occur. Okay, otherwise, if it is self-annealed, then the DNA replication will not occur. Okay, the strand will not get separated, and the replication will be stopped in that condition. Premature termination will be occurring. So this is the primer, and sliding clamp loader basically loads this sliding clamp onto this primer template junction. And the sliding clamp loader, after reaching to the 3' end, it releases the sliding clamp, and the clamp loader will again go to that position, okay, in its present position. And then the sliding clamp will recruit the DNA polymerase, okay, and DNA polymerase will synthesize the Okazaki fragment in this direction. So this is how the synthesis of the DNA occurs. This is called thrombone model of the DNA replication. Okay, so here you can see there is a here, for example, this is a primer. The tau protein will be recruited over here, and once it's reached the 3' end of the primer, it releases the sliding clamp. Okay, and DNA polymerase will bind in this region and synthesize the DNA polymerase in the Okazaki fragments. Okay, and the strain which is unreplicated currently, okay, which is a single stranded, is bound by the single stranded binding protein, so that it does not self anneal with the uh, top strand, okay, the parent strand, so that it prevents the religation. So single strand binding protein are bound in this region. So basically, the different parts of the replication works in to enhance the efficacy. The primase activity is stimulated by binding to the DNA helicase, so that the helicase moves at the speed of the helicase movement. Then only the RNA primer will get synthesized. Okay, and this actually ensures that the primer is made only at the replication fork only, and not the other region. So during the replication fork only, the primer should be made. If it is made in the other region, then there will be very problematic replica. There will be very uncoordinated mode of the replication occurring. Okay, so for coordination, primers are synthesized at the replication fork itself. Okay, and DNA 3 holoenzyme contacts the helicase via the tau subunit. So during the unwinding activity, so helicase DNA pol 3 holoenzyme contacts with the helicase, and that is how the clamp will be get. The DNA polymerase III will move with the helicase. Okay, and the sliding clamp helps with the DNA polymerase III enzyme processivity. So because of the sliding clamp basically helps in the binding of the DNA. So here you can see ring-like structure. So this ring-like structure helps in the form binding of the DNA. Otherwise, DNA polymerase III can also bind, but it will not bind as efficient as the sliding clamp. Okay. So if they incorporated a sliding clamp basically helps with the support for the DNA polymerase. So DNA polymerase, if the support is not there, then the DNA polymerase will, after synthesizing a few base pair, it will get removed from that. Okay, from that replication process. So sliding clamps helps in the supporting the DNA polymerase and thereby helps in the processivity. Yeah, so after the completion of these lagging strand, okay, the RNA primers are removed by the DNA polymerase one, which are present on the RNA primer, okay, and the gaps are filled by the DNA polymerase one. DNA gaps are formed by DNA polymerase one, and the nick are sealed by the ligase. So that is how you need to remove the primers as well, okay, from the lagging strand. That is an RNA primer. That is done by the DNA polymerase one removal as well as synthesis, and the phosphodiester bonds are formed by the DNA ligase. Okay, and the termination in replication act as a converging force. So, for example, the E. coli genome is like circular. Okay, so one helicase will move in this direction, another helicase will move in this direction. Okay, replication system, the replisome, and when they converge in the opposite direction, both of them over this position, the replication will get terminated. Okay, and this signals that the whole the DNA is getting got replicated efficiently. In the prokaryotes, in the eukaryotes, basically the chromosomes are linear. Okay, so linear chromosome basically during the end of the chromosome, the replication gets stalled. Okay, at the end of the chromosome, basically that is mediated to telomerase. So telomere sequence present at the end uh, uh, signals that the replication needs to be terminated now. Okay, at the end of the chromosome in the eukaryotes. Okay, so this was a like system, and for example, like when we compare, so all the system we talked about in E. coli, okay, the prokaryotes. So for example, in E. coli, the helicase which is present in DNA B, okay. So when we talk about the eukaryotes, the same helicase activity is the same, but we know as the CMG complex, that is CDC45, MCM27, and GNIS. So this is the helicase present in the eukaryotes, okay, compared to DNA B in the prokaryote. And the primase activity is done by the DNA G in the E. coli, but in the eukaryotes it is done by DNA pol alpha and primase. Okay, this is the enzyme. Single-stranded DNA binding protein is through SSB. Okay, SSB binds to the single strand. In the eukaryotes, it is an RPA. Okay, RPA protein. It is known as RPA protein. Sliding clamp loader, which helps in the sliding clamp loading and DNA polymerase activity. In E. coli, it is known as tau complex. In S. cerevisiae, that is eukaryotes, it is known as RFC. The sliding clamp in the E. coli, it is known as beta or DNA N. Okay, which binds to the prokaryote, which binds to the polymerase and helps in processivity. In eukaryotes, it is known as PCNA. And the DNA polymerase, replicative DNA polymerase in the eukaryotes, it is DNA pol 3 core enzyme. Okay, and in the eukaryotes, there are two enzymes, two DNA polymerase. Pol delta is for the lagging strand synthesis, and pol epsilon is for the leading strand synthesis. Okay, so this you need to remember. So in eukaryotes, basically there are three different polymerase. Another polymerase, okay, DNA pol alpha also helps in the during the formation gap sealing of the DNA replication. Okay, during the removal of the primers in the during the Okazaki fragment formation gap sealing. So there also another polymerase will be present, pol alpha. Okay, in the eukaryote. And in the eukaryote, generally the replication for progression is much slower. That is 20 to 60 base pair per second compared to the prokaryotes. In prokaryotes, around 1000 base pair per second is there. In eukaryotes, around 20 to 60 base pair per second. The replication fork moves in the direction. Okay, in the eukaryotes now as well, the DNA polymerase is not bound to clamp loader. So we talked about the DNA pol core enzyme. Okay, over here. So the clamp loader basically has a DNA polymerase attached to it. Okay, but in the eukaryotes, the DNA polymerase are recruited separately. It is not attached to this clamp loader protein. Okay. And Okazaki fragment also differs. In the prokaryotes, it is 1000 to 2000 nucleotides, and in eukaryotes, it is 100 to 400 nucleotides base pair. Okay, Okazaki fragment. So these are the some differences between the prokaryotes and the eukaryotes. Okay, so now we will move to some of the questions, the assignment questions. So we will solve. I mean, this is not an assignment question. This is a model question. So your assignment submission date is Wednesday. Okay, coming Wednesday. So after your submission only, we will discuss that assignment. Okay, in the next Saturday, next tutorial session. This assignment. So these are the some of the questions like from the previous year. Okay, model questions. So you can answer it based on the content which we discuss. So everyone can please reply. Okay, you can turn on the mic because if you type in the chat, then I will not be able to see because I'm in a presentation mode. Okay, so you can turn on the mic and you can answer. Okay, it will be beneficial to you as well during solving the assignment. Hello, is everyone there? Hello, hello. Okay, so what will be the option for first answer? DNA replication must occur accurately. So there are four options. What will be the answer? Correct option. What can be the correct option in this case? You need to answer, otherwise it is of no use. Okay, the questions. If you don't answer them, it's of no use. Okay, the first is to minimize the possibility of spontaneous mutation. The DNA replication must occur to minimize the possibility of spontaneous mutation, to reproduce and maintain the correct genome organization, and to ensure the duplication of all parts of the chromosome only once per cell cycle, and all of the above. So this one is correct to minimize the possibility of spontaneous mutation. Okay, during the base pair, during the RNase mechanism. Okay, proofreading mechanism will minimize the mutation. Okay, spontaneous mutation, and reproduce to maintain and reproduce the genome organization, and to ensure the duplication. So these all the three are correct. So option A, option D would be the correct. Okay, all of the above. So next question is, with the correct statement about the molecular structure of DNA. The two strands in the DNA double helix run parallel to each other. A binds to T and C binds to G. Base pair interact with each other by van der Waals interaction, and the four bases present in the DNA are AUGC. So out of these four, you need to pick the correct statement.
Option B, sir.
Option B. Yeah, other you can answer. There are very less students in the class. I don't know why, because in the previous two class, there like around 18 to 20 students strength was there. Do you know any reason why there are very less students in the class? Does anyone know? So the content what I discussed, so is it clear to everyone, or is it confusing? The explanation and all. So your feedback would be important in this case. So the discussion which we are doing, so is it helpful? Is it understandable, or is it complex? Okay. So option B is correct in this case. A binds to T and C binds to G. Okay. Others are incorrect. The two strands in DNA double helix run parallel to each other. It runs anti-parallel to each other. Okay, in the 5' to 3' and 3' to 5' direction. Two strand, leading and lagging strand. It runs anti-parallel. And base pair interact with each other by hydrogen bond, not van der Waals interaction. The four bases present in the DNA are ATGC, not AUGC. Okay, AUGC are present in the RNA, not in DNA. So this is third question. Pick the incorrect statement to the DNA replication. These are the four options you need to answer. You need to pick the incorrect one. So you can take 2-3 minutes and you can try to answer. Okay. Is the question understandable, or is it difficult? You need to reply, otherwise class will be boring. Okay, if only I will talk, it will not be interesting because I already know the answers. If you will not engage in this, then there is no point discussion. Okay, so DNA synthesis, the correct answer is option C. NTPs are utilized as a substrate for the addition of new nucleotide. Okay, so this is the incorrect statement. NTPs, DNTPs are incorporated. Okay, for the addition of new nucleotide, not NTPs. NTPs are utilized for RNA, not DNA. And DNA synthesis is occurred by extending the 3' end. This is correct. DNA polymerase catalyzes the formation of phosphodiester bond. This is correct. And during phosphodiester bond formation, pyrophosphate is released. Yes, basically the pyrophosphate, two phosphate group are released, and one phosphate is attached with the 3' OH. Okay, so this is the correct option. So each application origin are called. These are the four options. So in this option A is correct. ARS. Okay, so ARS is a site, the origin of application in the east. Okay, so this ARS site basically contains the ACS B1, B2, and B3 site. Okay, so on the ACS and B1, your ORC complex will bind, which is an origin replication complex. At the B2, your helicase will bind MCM helicase. Okay, and B3, where it is not shown, B3, other proteins will bind with help in the initiation and progression of the replication. Okay, so yeast replication origins contains an ARS site as a replication over origin sequence. Regulatory point in the animal cell, which is crucial for controlling the entry into the S phase of the cell cycle, is termed as restriction point, start point, entry point, and all of the above. The correct option over here is option A, restriction point. Okay, so in the animal cell, the regulatory point is called restriction point. In the prokaryotes, it is called the start point. So here you can see the explanation. So pick out the correct role played by the retinoblastoma in the cell cycle. So retinoblastoma is the transcription factor activator. E2F retinoblastoma bound complex activates E2F. Retinoblastoma is phosphorylated by G2M CDK, and active retinoblastoma inhibits the entry of cells into the S phase from G1 phase. So you need to pick the correct option out of this. So first option, retinoblastoma is a transcription factor activator. It is not an activator, it is a repressor of transcription factor. Okay, it represses the E2F transcription factor by binding to it. So this option is incorrect. E2F retinoblastoma bound complex activates E2F. No, it inactivates the E2F. So this option is also incorrect. Retinoblastoma is phosphorylated by G2M CDK. No, it is activated by G1 CDK. Okay, G1 and G1S CDK. So this is also incorrect. And active retinoblastoma inhibits the entry of cells into S phase from the G1 phase. Yes, so this is correct. Basically, the active retinoblastoma binds to the E2F factor. Okay, E2F site, and it inhibits the G1 phase S phase gene. Okay, inhibits the entry from the S phase from the G1 phase. And if it is phosphorylated inactive, then only the cells will move from the S to the S phase from the G1 phase. Okay, so this is option D is correct. Pre-RC consists of ORC, CDC6 complex, ORC and two copies of MCM complex, and CDT1 plus MCM complex. Which one of these is correct? Because option C is correct. ORC and two copies of MCM complex. Basically, the pre-RC complex is formed by the ORC and the MCM complex. Okay, so this is called pre-replicative complex, combining the ORC and MCM helicase, two pairs of MCM helicase. So option C is correct in this case. Activation of MCM helicase is carried out by DDK, which modifies the MCM helicase by acetylation, methylation, phosphorylation, or ubiquitinylation. Ubiquitinylation. So option C is correct. DDK basically phosphorylate. Okay, DDK phosphorylate helicase subunit and activates it. So DDK phosphorylate MCM helicase, and it will triggers the activation of enzyme helicase, and it will trigger the unwinding of the DNA, activation of the DNA. Okay, so option C is correct, phosphorylation. The semi-conservative model of DNA replication was experimentally demonstrated by which scientist? Messerschmitt and Stahl, Mira Troy, Charles Augustus, and Henry Blackwood.
Option A, sir.
Yeah, Messerschmitt and Stahl. So option A is correct. And in the semi-conservative model, basically, this is the explanation that in semi-conservative model, from the parental strand, the daughter strand will be equal proportional to daughter strand. Okay, 50-50 proportion. And during the second replicative cycle, there will be a proportion of 1 is to 3 is to 1. Okay, so new nucleotide and the parental. So in the first division, there will be a 1 is to 1 ratio of the parental and the daughter strand. Okay. And in the second division, the ratio will be 3 is to 1 of the daughter and the parent strand. Okay. So this is how the ratio will move. So if it is conservative, then there will be completely different ratio. Okay. So individual, it will replicate it. And in the dispersive model, you will find a completely new daughter strand with a slight replication in the slight DNA molecule of the parent strand. So these were the three kind of model proposed. But during the experiment by the Messelson and Stahl, they found the semi-conservative model to be prominent in that case. Okay, by the experiment. So during DNA replication, in which direction does the DNA polymerase synthesize the new strand? 5 prime to 3 prime, 3 prime to 5 prime, 3 prime to 3 prime, or 5 prime to 5 prime. Okay, so there is option A is correct. 5 prime to 3 prime direction. Okay, so this is the correct option. So DNA replication, 5 prime to 3 prime direction, the replication polymerase 3 will synthesize. Okay, so these are the total questions, model questions. So if you are interacted, then it will be of helpful for discussion better. Okay, but anyways, I hope you had understood the lectures. Okay, understood the discussion. If you have any doubt, then you can ask. We have still little time, so if you have any doubt in any of the concept, you can ask. Otherwise, we can end the session. In any of the slides, if you have any doubt, then you can ask. Okay. Okay. So thank you, everyone. If don't have any doubt, then we can end the session. And thank you for joining.
Thank you, sir.